Hi, everyone. This is Sheri Wilcox. I'm the Vice President of Probe Development and Bioinformatics at Nautilus Biotechnology, and I am excited to share with you the presentation I gave this year at the 2025 US HUPO conference on characterizing the protein binding properties of multi-affinity probes for quantitative proteome analysis. So first of all, why do we care about single molecule intact protein detection in the first place? We feel that when we are able to interrogate an entire intact molecule, we get the most accurate and specific protein measurements that can be taken. In addition to that, we have the opportunity to collect information on precise proteoform measurements within a given molecule. Anytime we're talking about a single molecule platform, we're able to count up the quantity of any given protein or proteoform by its identification, and therefore it is as quantitative of a measurement as you can get and sensitive down to those individual molecules. So the Nautilus platform, just as a brief overview, a lot of you have probably already heard other talks about the Nautilus platform, so I'm just going to give you a high level overview um, of the various steps of the assay. We start out by preparing a library of the single molecule proteins that are present in the sample, and then deposit those onto a flow cell. Then we can either work in a targeted proteoform mode or in a broad scale mode with our proprietary multi-affinity probes to iteratively bind reagents to those displayed molecules. And I'll show you more about this in a video in a moment, and record binding events using our instrument. Then we take those binding events and we're able to run some proprietary machine learning software that allows us to identify and quantify the proteins and the proteoforms in that particular biological sample. So this is a video that describes a little bit of how the platform works, whether we're using our binders that recognize proteoforms in a targeted manner or our multi-affinity probes that I'm going to talk about a bit more in the following slides. We start out with nanoparticles that have attached to them a single protein. That nanoparticle is then able to land in defined positions on our flow cell. Then we flow across our binders, which have fluorescent tags on them, and we record the fluorescent signal at each of these positions on the flow cell. And so you get these kind of starry sky patterns. We then remove that and rinse it off and flow a different binder across for the next cycle. Again, we're going to take a picture and record where the fluorescent signal was. And then we're going to take another one. We just keep rinsing and repeating. So we have a binding event, we record what was bound, we remove that, flow something else across, record what's bound, and then remove that. So we're able to then stack up a lot of these images, about 200 to 300 in our broad scale application, in order to identify what proteins are bound in each position. We liken this to the game Guess Who. So instead of walking around to try to figure out this animal by asking, are you each of the individual animals that are the choices for this game, Guess Who? That takes a really, really long time. And we found that instead of asking that very specific question, if we ask general questions, we can get to the answer a lot quicker. So in the case of these animals, if we ask about the properties like, are you a mammal? Are you a large mammal? Are you a cat? Are you a woodland creature? You get to the fox a lot faster than if you went around and asked about those individual animals. Same thing happens with our multi-affinity probes. Instead of going through and asking the question with those binders that I showed you in the video, instead of saying, oh, is this protein in this position CRP? Is this protein in this position IL-6? No, we ask about the presence of short stretches of amino acids that are present in a lot of different proteins so that we can build up these patterns. And so we go through these series and we have binding events. In this case, we're showing an example. This is synthesized data. This is not actual data here, but we're showing an example where perhaps you'd had a binding event for a lysine, valine, lysine, and it lit up. The next cycle did not light up, so we wouldn't record what the recognition site was for that particular probe. when we're trying to identify a given position. And then in the third one, perhaps it's an asparagine, methionine, serine. And so you build up this pattern, not in order in the protein sequence, but just anywhere across the sequence. And we're able to deconvolute that against the FASTA file of the human proteome. You might ask, well, can you even make probes that can bind such short stretches of amino acids? And the answer is yes. We have built pipelines that can generate a lot of probes that typically target about 3 amino acids embedded in some kind of flanking sequence. The primary pipeline we're using right now is phage display, where we are panning to get SCFVs and then convert those SCFVs into IgGs. We then take the candidate probes forward and we characterize them with a number of assays. One, we care about the kinetics of our binders, and so we use BLI, biolayer interferometry, to characterize the binding. So here are two different sensograms from various candidate probes, and you can see the association phase of the binding and the dissociation, and you can see that we have some events that have slower association and slower dissociation, or we might have very fast association and fast dissociation. So we are going to choose which ones of these fit the best parameters for our assay. In this case, this is about a 3 nanomolar KD with a half-life of greater than 1,000 seconds. And the lower one is more like a 300 nanomolar KD and a half-life that's less than 10 seconds. So that's going to be less useful for us on our platform. Furthermore, we want to understand exactly what's bound by our probes. Now, I said that we're typically targeting about 3 amino acids, but we don't really want to be a perfect specific probe for those 3 amino acids, or we'd have to do this 8,000 times to cover all of the amino acid sequences. So instead, we actually are looking for very reproducible motifs that have some flexibility in those positions. So this is an example of a probe that we developed by panning against the 3 amino acid peptide tryptophan, asparagine, lysine. And we use a number of tools to then characterize, we call it profiling, exactly what the epitope is that's recognized by this probe. And we've typically used FIPSeq, which is phage immunoprecipitation and sequencing, and peptide arrays. And so this is a set of data where these are the top sequences that were captured by this particular probe that was panned to bind this WNK sequence. And what you can see is that each of the peptides that are captured do indeed contain essentially WNK, but there's some variability. In this case, instead of the lysine, there's a serine. There are other times where a phenylalanine can be present instead of that tryptophan in the first position, or even an arginine instead of the lysine. And this motif down here just shows us the conserved consensus sequence for what's bound. I also find it reassuring. These are actually human sequences, human peptide sequences here. And these top hits, you'll see they're very similar. And that's because they are actually isoforms of peptides or peptides from isoforms of the same protein. And so this also lets us know kind of what's the overall proteomic coverage that we should expect from a given probe. Now, local plots are great for us as humans to look at with our eyes and try to understand, but there's a lot more nuance into the actual binding of a given probe than just what you can see in a motif. We've also looked at the variation of the N-terminal flanking and the C-terminal flanking sequences to the motifs in these enrichment types of assays. So for example, when we have the canonical tryptophan, asparagine, lysine that was the original panning peptide in the first place, if we have an N-terminal asparagine, we actually only capture about 35% of the peptides that had that sequence. If we have an N-terminal glycine, it's about 80%. And so it varies a lot depending on the surrounding sequence, and that's okay, because we can use deep learning methods in order to incorporate all of this data into the protein binding probabilities that we're going to map against a FASTA file of the entire human proteome. Now, we really care about how well they bind to proteins. And so one of the ways that we then assess this is using Western blots, because that's an easy way for us to look at binding to proteins. And so these are 3 different probes. Each blot is a different probe binding to 3 different proteins. And what you can see is that there are specific probes is probe 1 binds very well to vitronectin, probe 2 binds to vitronectin and a little bit to plasminogen, and then probe 3 binds to lactoferrin. So as you can see, we are able to demonstrate the binding not only to peptide sequences that contain the expected motif, but also to a full-length protein that has that motif. Now, the plasminogen band, I'm sure you're looking at and you're saying that's pretty weak. And you're right, that's a very faint band because it's not a perfect motif here, but it is one that we expect to bind, and we can calculate the binding probability. In addition, we can take these same probes and these same proteins and run them on the actual Nautilus single molecule platform and measure the binding there as well. So here we're looking at the detection values on a flow cell for these, again, 3 different proteins plus a null nanoparticle that doesn't have a protein bound to it at all for these different probes. And just as we saw in the previous slide with the Western blots, probe 1 binds to vitronectin. And so you can see probe 1 is binding to vitronectin and not so much to lactoferrin and plasminogen. Probe 2 is able to bind very well to vitronectin and a little bit to plasminogen. So again, here's probe 2 binding to vitronectin and a little bit to plasminogen. And then probe 3 is binding very nicely to lactoferrin and not to the other 2 proteins. And here's the intensity of probe 3 binding to lactoferrin at the beginning of our assay, and then about 40 cycles later in the assay, binding again. So you get to see the reproducibility of that. Overall, we can then take these types of probes and extrapolate out how many cycles we need to run in order to identify about 95% of the human proteome. And we expect to need about 200 to 300 different probe binding cycles in order to essentially quantify 95% of the human proteome. Just to remind you again, the beauty of the single molecule assay is that we get to couple identification to quantification because you have all of these individual nanoparticles. And each time you identify what's the most likely protein, you can then just add those up. Here, P53 is highlighted. And so you count up how many times P53 was the most likely identity. And that's how you can quantify the protein in your sample. We believe the Nautilus platform is going to be able to unlock insights across all kinds of applications, from biomarker discovery and validation to target discovery and validation, mechanism of action, toxicology, and whatever research question you'd like to bring to us. Now, I realize you guys are joining this late, and so you don't get to participate in our scavenger hunt that we had for a Nintendo Switch. I'd encourage you to join us at any of the meetings that we participate in the future, because we oftentimes do these scavenger hunts. And a lucky winner who gets all of the clues of these short amino acid sequences at our various talks and posters gets to come by and win a Nintendo Switch that is branded just like Nautilus. Thank you for your time, and reach out if you would like to have any more information about the Nautilus platform.